Without further ado, let me just quickly introduce our keynote speaker for today, Dr. Hina Chaudhry. You can read her bio online. She's highly accomplished. Typically, what we in our previous webinars we gave each presenter a topic. We felt that Dr. Hina has done so much in so many different fields. Why don't we just keep it light? We'll keep it short on presentation, but more on Q&A session. So we would really like you to post your questions in the Q&A panel, and at the same time, we will ask questions with Dr. Hina. A lot of individuals have already sent out their questions, just going through her bio and what she has done, and her successful achievements and whatnot. The other thing that I really wanted to point out is the Sitara Imtiaz. I did not know that. I was just looking at her bio and I found that out. So I think we would want to hear about that as well as we go along into the session. So now, without further ado, Dr. Hina, I will make you presenter, but at the same time, if you want to just maybe start talking about your thoughts here and enlighten us, please.
Okay, thank you very much, Zahira. I want to say hello, assalamu alaikum, everybody. Really happy to sort of share some of my journey with you. My slides are a little bit on the technical side. I've cut out many of them and I'm just focusing on the key slides. So what I'm going to do is go through the basic slides, and at the end there's a couple movies that on this newer Mac that I have, they're not playing appropriately inside the slide presentation. So I'm going to save two of the key movies for you at the end after the slides are done, and then feel free to ask me questions. I'll throw in a few things about how I got started on this path. So I'm basically, I'm a cardiologist. I'm a clinical cardiologist. I do see patients. I do patient procedures for the cardiology department at Mount Sinai Hospital in New York. But I'm also an associate professor. I've been told in the midst of being promoted to full professor now soon. And I run a research program in cardiac regenerative medicine at Mount Sinai. I'm also the founder of a biotechnology company called Venture Nova, and that is a biotech startup to commercialize the therapies that I've developed in my research over the past now almost more than 15 years, to bring them to market and so that patient lives can actually be saved based on the research that I've been doing since I was a young trainee in my 20s. So my opening slide is actually the actual picture of an actual heart muscle cell. It's shaped like, the Valentine's heart, but this is the way it was staring up at us in the microscope. We were the first to show that you can induce cell division and differentiated, what are supposed to be differentiated cardiomyocytes. So this is sort of a microscopic analysis, and the green fluorescence is a structural protein inside the cardiac muscle cell. I'm going to refer to these are cardiomyocytes, are the actual cells in the heart that beat, because there are a lot of non-cardiomyocyte cells in the heart. The cardiomyocytes, although they comprise the bulk of the volume and the mass of the heart, they comprise about only 30% of the number because they're so large. There are a lot of other smaller cells, in the extracellular matrix and the vasculature and everything. But my work focuses on the cardiomyocyte and making it divide, or other types of, or stem cells that can actually differentiate into beating cardiomyocytes, because that's the key to regenerating the heart. We have to be able to build new cardiomyocytes in the damaged heart. So this is an actual picture, like I said, of an actual cardiomyocyte. The blue staining signifies the nuclei, and the red staining is a particular protein that undergoes phosphorylation during cell division, phosphorylated histone H3. So basically, lower vertebrates, such as here we have the North American red-spotted newt and the zebrafish, they're able to regenerate their hearts. And I think that understanding the mechanisms by which these animals regenerate their hearts was very helpful to me in trying to design strategies for human heart regeneration. In both of these animals, the heart regenerates through proliferation of the native cardiomyocytes. It was not a stem cell approach or so. Their hearts don't have stem cells. They have cardiomyocytes that can sort of de-differentiate and divide and then differentiate back into mature cardiomyocytes. So this is really an approach that inspired me. It's worked very well for me. This is a basic diagram of the cell cycle. You see that cyclin A2 is regulating the cell cycle, the cell division cycle. Two key transitions, the G1S transition as well as the G2M transition. And because it's the only cell cycle regulator that guides the cell cycle through the two key transitions, I felt that it was very important to study. It's also the only cyclin that undergoes complete gene silencing after birth in all mammalian species. And that's what also why I felt that this was going to be a key player in cardiomyocyte division. And my early work as a postdoctoral fellow that was published back in the early 2000s had shown that it is the master regulatory switch that guides cardiomyocyte cell division in the embryo, in the mammalian embryo. It is turned off after birth, and we can turn it back on, and cardiomyocytes will divide. When we turn it back on, all other cell cycle regulators have increased expression in the cardiomyocytes, and therefore we knew that this is basically the key driver of cell division in cardiomyocytes. So this has been the first strategy I've developed that is now up for approval at FDA, is a gene therapy strategy that administers cyclin A2 into the diseased heart. So I'm skipping over about more than a decade of work in small animals and getting right to the large animal work. So we had used a gene therapy approach in the pigs we had studied after we infarcted them. And just a funny joke is that because I'm a Muslim and my first author, Scott Shapiro, was Jewish, he nicknamed these pigs his kosher pigs, and I called them my halal pigs, just because, we're studying them to cure disease. I actually, because I was a principal investigator, didn't actually handle them myself. But it's a very important step when you want to get FDA approval for a therapy that you have to test it in a large animal. And for heart cardiac therapies, the standard is usually the pig model. And so this paper landed in Science Translational Medicine. This was published in 2014, and we got a lot of press. As you can see, there was a Scientific American article. It was highlighted in the TEDMed blog because I had presented my approach at TEDMed a year before that, and Market Watch, Fierce Biotech, and BioCentury, some of the biggest sort of news media about scientific and pharmaceutical developments had carried this story. It was a very exciting time. And then after that, so this is just a summary. I won't go into detail about what we did, but here we saw a 55% increase in the numbers of cardiomyocytes in the peri-infarct area in the pigs, and we had ejection fraction. Under panel C, you can see ejection fraction improved markedly. The ejection fraction is a measure of the contractility of the heart. Improved in the pigs that were treated with cyclin A2, whereas it went down in the pigs that received the control virus, which is our control group. And then after that, we started studying the gene therapy approach in adult human cardiomyocytes. And I have to say, it took us probably 2 years to develop these techniques that allow us to study this process. No laboratory in the world has been able to culture adult human myocytes for up to a month, which is what's required for these studies. And we had to develop all the techniques and the tools in my laboratory in order to even study this process. And so here we see in the first panel a typical rod-shaped cardiomyocyte. It's from a 55-year-old man. This red fluorescence you see is a tool we developed to highlight the living sarcomere of the cardiomyocyte so we could follow it during live imaging under microscopy. The green is another fluorophore, a green driven by a cardiac promoter, troponin, to prove to us beyond a shadow of a doubt that this indeed was a cardiomyocyte. And then we turn off the green channel for the imaging because the green light can kill the cells. And you see over a period of 70 hours, and this is also one of the movies that I want to show at the end, in response to the gene therapy with cyclin A2, this human cardiomyocyte undergoes 2 rounds of division. The first one you can see at 45 hours, and it's finished by 50 hours. And you look at the bottom panels at 65 hours, it's undergoing a second round of division. This has never been done before. This paper is in review right now at a very prestigious journal, and we hope it'll also be out before the end of this year. But this has been impossible. I mean, in human history, nobody's shown that adult human myocytes from somebody as old as 55 years old could be dividing like this. So we're very excited about this. And so this is actually the PowerPoint of the movie, and it's, like I said, we'll have to play it for you at the end. And now I'm going to switch gears a little bit. I also study stem cells, and we have a new paper coming out before the end of this year on a very novel discovery of a type of stem cell that can be isolated from the placenta. And it can be found in the end of the term human placenta. So as you know, placentas are discarded in labor and delivery rooms around the world. So this is a limitless source. We would not even offend the Vatican because we're not taking embryos to isolate embryonic stem cells. This is a type of cell that is coming from the outer layer of the blastocyst. And then in the end gestation placenta, there is this small population of cells that are left over from that early developmental stage. And we have found that they can form beating cardiomyocytes in a petri dish and that they can repair the heart in a mouse model of myocardial infarction. And my lab had pioneered the work of showing cells can transfer from the fetus through the placenta to the injured mother's heart, so that the mouse shown at the bottom. We had published a first paper in 2012 where you see Kara et al. Kara was a graduate student, Rina Kara, in my laboratory, and we published an early paper back then showing that fetal cells naturally sense injury in the mother's heart. If we had injured pregnant mothers in mice at mid gestation, and we could track fetal cells coming to the injured mother's heart, repairing the injury, and they zoomed only to the injured portion of the mother's heart, and they did not go to non-injured organs. So then what we developed after that was we developed a strategy whereby so we saw these markers, and I won't go into detail. We saw a whole bunch of markers on the cells that were homing to the heart from the fetus, and we found that CDX2 was prominently displayed on about half of these cells. So if you look at the history of what CDX2 is, it's involved in this trophectoderm layer in the early blastocyst, which is the part that implants in the uterine lining. And like I said, at end gestation in both mouse and human, we are able to find a reservoir of these cells. And so we had found a way to genetically label them. My postdoc, Sangeeta Batake Madatil, has developed a way where she labeled the cells green. She has studied them in vitro in a petri dish and in the in vivo injured heart model as well to see if they repair the heart. And so this is just a strategy where she developed, where she engineered mice. If you look at the bottom part in the middle, this mouse, she engineered the mouse so that a green fluorescent protein is knocked into the CDX2 locus. And that's what allows us to isolate these cells. And like I said, my movies unfortunately are not playing, but if you look at this slide, this is really cool. There are so many green cells here. These all arose from a single clone, and they all beat in the petri dish, which has been a major stumbling block in the cardiac stem cell field. Is it's really hard to find stem cell types you can isolate from the adult heart that will actually beat in a petri dish. The only kind that actually do this are embryonic stem cells and induced pluripotent cells, and they have a lot of technical challenges with the use of those cells for heart repair. So we're very excited about this. This has shown that it produces, cardiac structural protein. There you see the green nucleus, and the upper left panel by the bottom right, you see that it has differentiated into cardiomyocyte. You can see it's embedded in a cardiomyocyte that even has the sarcomeres that are known to be present in cardiomyocytes. The proteome, we've studied the proteome of these cells, and we've shown that they retain all the stem functions of embryonic stem cells, but they have additional features in survival and in homing and in evasion of the host immune surveillance system. So they make an ideal, what we call allogeneic cell therapy strategy, which is what you can take cells from one organism and transfer them to another organism of the same species, and they will not be recognized by the host immune surveillance system. So therefore, we know so far they represent a unique multipotent cell type in the placenta. They form spontaneously beating cardiomyocytes in vitro, and they're immunologically naive. So this is an ideal strategy if you want to one wants to use for cell therapy, and now we have tested them in a mouse model. So what we did here was we took male mice and we infarcted them. These are genetically naive adult male mice. And we isolated the CDX2 cells from the placentas of the female mice, which, my postdoc had developed that genetic model where they're tagged with the green protein. So we isolated them from female placentas. We administer them as shown here in the tail vein of the male mouse. And we did MRIs 1 and 3 months post-treatment. We sacrificed the mice 3 months post-treatment to look for green cells and where they end up. And lo and behold, we've shown that the cardiac function of the mice that were treated with the CDX2 cells improves dramatically at both 1 and 3 months. On the left is a PBS control. There's no change in the group that was treated with PBS. In the middle are the control cells where we use CDX2 negative cells from the placenta, and we see no improvement in cardiac function. So you see on the right side with the CDX2 cells, we see significant improvement. And here is on slide 24 now, showing you that these green nuclei you're seeing are the CDX2 cells because they're labeled green, remember? And then we use markers for cardiomyocytes, which is red, and the blue are markers for the nuclei. And then we show a merged one, and it's a blow up of that on the right side. You can see this nucleus, which is expressing both blue and green. So we know it's a nucleus. We know it's came from a CDX2 cell because it was green in the first panel. And it's buried inside a completely differentiated cardiomyocyte because you can see the sarcomere structure of the cardiomyocyte. And at the top and on the right, you see we've done what's called Zstacking analysis. We do a 3-dimensional analysis of the cell to show that the nucleus is indeed buried in the cardiomyocyte and it belongs to a cardiomyocyte as opposed to being outside a cardiomyocyte cell in a different type of cell. And here we're also seeing formation of blood vessels. You can see the linear structures form and the antibody staining for the typical endothelial cells, and that you can see from the first panel, there were green cells lining that, so they're coming from the CDX2 cells as well. So now we have what's an ethically compliant and potentially multipotent cell source for allogeneic cell therapy for cardiac, and we're hoping other types of organ repair. Because these cells are able to form multiple types of cells, I'm very hopeful that we can apply them to other types of organ repair other than cardiac as well. I'll just show you the rest of the slides really quickly. This is just some major publicity we had about this cell therapy approach and a number of new publications. We have a lot of my collaborators that are at Columbia, where I started my faculty career, and at Mount Sinai and Rockefeller. David Alice just won the Lasker, which is a predecessor to the Nobel Prize. He's the one who's taught me everything I know about gene silencing. So I'm very thankful for this incredible investigators all over New York at many institutions that have aided me in my work. This was a picture of my laboratory, which is mostly the group that contributed to the cyclin A2 gene therapy work. And then this picture shown in the middle is Kelly Wright, who's a Fox TV anchor. He filmed a special in my laboratory, an episode of his series called Beyond the Dream, about my work. And then to his right is Sangeetha, is the postdoctoral fellow who has spearheaded all of the cell therapy work, the CDX2 cell work in my laboratory. And to his left, Jesse Yoon is a medical student at Mount Sinai who's also contributed to the CDX2 story. So I have my lab sort of studying 2 different aspects of regeneration, and I wanted to tell people, don't be just, don't be completely buried in work. You have to get your mind off work. You have to do some exercise, because that actually creates new brain cells in the brain, we now know. And thanks for all my work. Of course, my dad, this is a picture taken towards the end of his life, and on the right is my mom the day I was getting the Sitara and Imtiaz. As you can see, I'm in the background fiddling around with the metal, and she's just looking like the happiest I've ever seen her. And that's the end of my slides, and I was hoping that we could show you the movie I'm talking about, because that will also help answer your question, Sarah. So this is a representation of what we are doing. We'll be administering the gene therapy, the first approach I described, in the cardiac cath lab, infusing it down the coronary tree through simple, procedures that are present in the cardiac cath lab. These golf ball shaped objects you see are the carriers of the virus, which is being transmitted. The green particles, they diffuse through the heart. We've seen that coronary diffusion is very high. Virus particles will distribute through the myocardium and induce the cardiomyocytes to divide, and that's how you'll heal the infarction zone. So that's just a sort of a digital representation of what our therapeutic approach looks like with the gene therapy.
Why don't we start with our Q&A as well, so that in the interest of time we don't